All right. So right now we're going to be talking about enzymes, and enzymes are proteins, right? And we learned about proteins, and we talked about biomolecules. And so you may be looking at this picture right here and be asking, well, what the heck is that? This is an enzyme right here, and an enzyme is made of proteins, and these right here are just a bunch of polypeptide chains. Remember, we talked about that when we talked about biomolecules, and that a lot of times a protein is made up of different strands of polypeptides, right? And they're kind of put together. And here, this is why they're different colors, kind of showing you that they are different proteins kind of connected into one larger protein, right? And right here, you have what is called an enzyme. And these guys are usually called like the workhorses of the body. And you'll see why in a little bit. And so, what are enzymes? Enzymes, like I said earlier, they're proteins, right? And a lot of times I mention that a protein will end in the word "in," but if you're talking about specific proteins like enzymes, if it's an enzyme, it's going to end in "ase," right? So, like sucrase, lactase, all of these that you see ending in "ase" are a pretty good indication that it's an enzyme. And so they act as catalysts to accelerate a reaction. So if you're thinking about a catalyst, if you're trying to define that, if you're a catalyst for something, it just means you speed up that process, right? So if you're the catalyst of the party, whenever you show up to a party, you get it started, right? That's kind of what a catalyst is, right? And so it speeds up a reaction that happened in your body, and so not permanently changed in the process. So that's the thing, is you're going to see that enzymes, I don't know, to me they kind of remind me of like that Pac-Man shape. I don't know if you are familiar with Pac-Man. Kind of looks like Pac-Man, the way the mouth opens, it can fit certain objects in there called substrates, and you'll see right now. But basically, when this happens, the Pac-Man keeps its shape at the end, right? It's not going to change into some different shape other than that Pac-Man shape. And they're not all necessarily looking like Pac-Man, but that's just kind of how I think of it. That it'll just always come back to that shape, right? Even after a reaction happens, it comes back. And so here you kind of see that. And I don't know if you can kind of visualize a little Pac-Man head right there, but this right here is an enzyme, and something is going to fit in that little space right there. And that space is called an active site, and we'll see that in a little bit. And you may hear me repeat some things over and over, but again, that's what learning is, right? So if you're like, oh, I know that already, quit saying that, then that's good, right? It's a good thing that you know that already. And so again, this is that little Pac-Man shape called an enzyme, and that little whited out part right there is what is known as an active site. And let me just take the opportunity right now to just kind of put this right here, and we'll see this again in more detail. But this right here would be the active site. And so basically it'll fit something in here, and it'll either fit two things and connect them and turn them into one thing and then spit it out, or it'll take two separate things, kind of take it into that active site and then spit it out as one solid thing. So usually it builds up or breaks things down, right? But the important part is that it's not going to permanently change its shape when it does those things. All right. Oops. And so here you kind of see that what I was talking about. Here you have that enzyme. And again, not necessarily Pac-Man shaped, right? But you see right here, this is an enzyme and this is the active site. This thing right here that I was talking that I had sucks those things into the active site, those are called substrate, right? So a substrate, like you see right here, it's one big piece. And what happens is it comes in here into the enzyme. And when they're connected like this, y'all, it's got some really fancy name called the enzyme substrate complex. It's just what it sounds like, right? It's got an enzyme, this blue part. It's got the substrate, which is this triangle-ish looking thing, and this oval thing. And so basically, a complex is like a big structure, right? And so this is just an enzyme and the substrate when they're hooked up together. And this is a complex, right? And so when they're all together, enzyme substrate complex. And here you see this arrow degradation. To degrade something just means you break it down, right? If you degrade a person, it basically means you're breaking them down, right? You're kind of telling them off or things like that. And so to degrade something means to break it down. And so here it's degrading the enzyme, this blue part is degrading the substrate into two different products, right? So it just took that one shape and broke it up into two. Right. And here you see it spits them out, but look at the enzyme. It kept its shape. Right. And so here you have synthesis. The opposite of degradation is synthesis. Right. So where you're breaking down here, you're actually building up here. Right. So remember, y'all, this is what your body does all the time. It's always building up building up or breaking down, right? That's kind of what metabolism is just building up, breaking down, building up, breaking down. So, there's so many different processes where this happens in your body that it's good that these enzymes don't get used up, right? Because it lets them work harder and more efficient. If you had to recreate an enzyme every time, kind of be a pain in the butt. But anyways, here you have the substrates, right? And they're two individual ones. And so this enzyme here will kind of take them into the active site. Remember, the active site is just kind of where they hook up with the enzyme. And then right here, it goes through synthesis, which means it just builds it up into one larger product, right? So these are all chemical reactions, y'all. Chemical reactions are building up, breaking down, building up, breaking down. And that's what metabolism is. And so chemical reactions, it's what I was just saying. It's they're always building up or breaking down. They have reactants that go in, they react, they go in, and then products that get spit out. Right. And so chemical reactions, though, they need an initial input of energy. Right. That's the activation energy. So, like striking a match, that is a chemical reaction, right? When that match combusts into flame. But you need something to get it going, right? So that means you kind of need to put in the energy to strike the match against a box and then get that flame going, right? The same thing kind of happens in our bodies where there's a reaction that needs to happen, but it needs a little bit of a something to get it started, right? A little bit of a spark to get it going. And so, what is really cool about enzymes is that they reduce the amount of energy that a reaction needs to get started, right? So, right here, you see the reactants, right? That's what goes into a reaction. And then at the end, you get the products, right? That's what comes out. And so it's kind of what we were seeing when we were looking at that enzyme building up and breaking down right here, right? And so in order for that to happen, it needs that energy. But what's really cool is that the enzyme is that catalyst, again, that speeds up a reaction that reduces the activation energy right here. And so instead of having to climb this huge like hill right here, use this energy, it happens a lot faster. So we'll see that right here. Enzymes work by weakening bonds, which lowers the activation energy, right? So instead of having to use all of this energy right here to for the reaction to start, it reduces that. So now there's not much of a hill to climb, right? So it makes this happen a lot faster. And here's an enzyme catalyzed reaction pathway, which sounds really complex, but again, it's just what we talked about, right? The enzyme, which we saw as that protein, catalyze means you speed up a reaction, and this is just the path that it takes, right? So again, enzymes work by weakening bonds, which it lowers that activation energy, right? So that's the important part to remember. Enzymes lower the activation energy. And so here you kind of see that without an enzyme, and I'll move to that right now. You're going to see kind of this slope happening. And so this is without an enzyme. But look what happens with the enzyme. You use a lot less energy right here. Look how much was reduced. So instead of having to climb this hill, use this much energy, an enzyme makes it so that it doesn't, right? And it speeds up the reaction. Like it says right here, the progress of the reaction, it happens a lot faster because it doesn't need to use that much energy because the enzyme has reduced the amount of energy it has to use. Right. And so here we see a little bit more of that. Again, it's repetition, right? But it's really good to cement these things in your brain. So activation energy of a catalyzed reaction is so much less, right, than if we would have had to have done it without this, the catalyzation of the enzyme, right? So here you see the new reaction pathway, this dash line right here, which again is a lot lower than if we wouldn't have had that enzyme. And it speeds up that reaction quite a bit. An enzyme controlled reaction proceed 10 to the eighth power to 10 to the 11th power times faster than non enzymic reactions, right? So without this enzyme, again, this eight right, or I don't know why I said again, I haven't mentioned this before, but this eight right here basically means that's how many zeros you would add on to this number right here, right? And this 11 kind of means or it does mean the same thing, that that's how many zeros you would add on to this number. So imagine that, right? It takes a lot longer for a reaction to happen without those enzymes than with them. And we wouldn't be able to function or to live right without these enzymes in our body, because by the time these reactions happen, we would have already passed away, because we need these reactions to happen a lot faster to keep us alive. And so that's the thing to remember too. Enzymes are specific. That's why I mentioned earlier with the names, right? Luke, sucrase, what did I say earlier? I've already forgot, but I'm going to show you some examples right here. I was trying to think of the ones that I mentioned earlier. They're specific for what they catalyze. And a lot of times, because of that, they're giving very specific names. So there's so many different reactions happening in living things that yeah, you do have to be very specific when you're trying to label specific ones: sucrase, lactase, and maltase, right? These are all different enzymes. And again, y'all, enzymes will either be building up these uh, what's the word, substrates. They'll either be building them up or breaking them down, right? So in the case of lactase right here, for people that are lactose intolerant, they have their enzyme doesn't break down lactose, right? So when people buy lactose free milk, they're basically already adding that enzyme inside of the milk so that it breaks down lactose, so that people who cannot break it down in their bodies, the enzyme does it for them, right? So usually when you have lactose free milk, it usually tastes a little bit sweeter, because remember, when you break down bigger sugars into smaller sugars, it tastes sweeter, right? So here, like in the case of lactase, it is an enzyme that breaks lactose down into smaller sugars. But again, for people who are lactose intolerant, they can't do that. So that enzyme is already put into the milk for them, so that it does that for them, right? So they don't have to worry about some of the issues that come with being lactose intolerant. And so, again, just remember, these enzymes right here are either building up or breaking things down, and very specific to what they do. So in the case of lactose, lactase is that enzyme. Right in here, you see again the enzyme, which is that greenish thing. Then you see the active site, which is where the substrate goes into, and then you see the substrate there, right? It looks like in this case, it would probably be um, a breaking down reaction, just from what it looks like. And let me just add this right here, y'all. If you're talking about a breaking down, so breaking down right here is what is known as catabolic. Let me kind of expand on this a little bit. There we go. And then when you have something that's building up, that is known as anabolic. And that's why they call them anabolic steroids, right? Because they're supposed to help build muscle. And something that is catabolic means it's breaking down, right? So these are the kind of reactions that, I guess that's a good place. Let me just close this down. So that's basically, all the reactions that are happening are either catabolic or anabolic. And so here again, we have the enzyme. Remember that the substance, a reactant, an enzyme acts on is a substrate, right? So the substrate is basically the thing that would go in here into this active site. And so here's that substrate right there, and it joins here at this active site. So remember, this part right here is going to be the active site. And so a restricted region, which is what I was just talking about, right? This is a restricted region. So basically it means it's only meant for one thing right here. The restricted region of an enzyme is where it binds to the substrate. So again, the active site right here. Let me kind of put this in the same color as this right here, which kind of this yellowish color. So right here, remember, the active site is this guy right there. All right. And so the substrate, remember, it goes into this active site. Remember, when they're all together, this is what's known as the enzyme substrate complex, right? And so remember again that it is restricted because it's only meant for this certain type of substrate right here. Enzymes are very specific, and it's kind of interesting. That's what we're going to see right now, as how it kind of fits together, almost like in a lock and key type of way, right? And there is this hypothesis called the lock and key hypothesis, which says that there's a fit between the substrate, right, which we saw right here, this yellow thing, and the active site of the enzyme, right? It's exact like a key fitting into a lock. And so here you call this an enzyme substrate complex, right? It's just what's formed right here when you have them all together. The enzyme, the substrate, and it's one big complex, right? So the enzyme, they're formed, and products have a different shape from the substrate. Whoops, a little bit too quick. Products have a different shape from the substrate, and products are released from the active site, right? So again, it's kind of like the Pac-Man. It swallows them up and then it spits them back out. Rather, it'll take two separate things or multiple things and turn it into one, or it'll take one thing and kind of separate it into different parts. And so the products are released from the active site. So here you kind of see that lock and key hypothesis, where the substrate is designed very specifically like a key to fit into a lock, right? And so when they come here, you see you have the enzyme substrate complex, and here you have the activation energy, which is lowered. Remember, without this enzyme, it'd probably be like up here somewhere, but we do have the enzyme, right? So it's not as high the activation energy that it needs. And so right here, when it goes through the reaction, you see it broke down this pink substrate into two green substrates, right? So this one, you would call this catabolic because it's breaking down. Remember, it's either building up anabolic or breaking down catabolic. And so that's what it's doing right here, right? It's taking one thing and breaking it down into two separate things. And so here you have the products, right? Remember, the products are just what comes out at the end. And here you have two different products. And the enzyme can be used again. And that's what we were saying, right? That's what's good is they can just kind of hang out here and do this over and over and over again before they are replaced, right? So it would not be very efficient if they kept breaking down every time. So it's good that they don't, right? You can be using them again and again. Here, let me kind of close this down a little bit. And then there's the induced fit hypothesis. This hypothesis says that the substrate binding slightly changes the shape of the enzyme, right? So that it's not exactly like here, like the lock and key. Look how exact this is right here. The active site is designed exactly or shaped exactly like the substrate. So when it goes in, it fits just perfectly in there. But what the induced fit model is saying is that the enzyme, it kind of shapes itself to fit around the substrate right here, right? So right here, it's not matching this right exactly the way it is over here. But when they do meet, the enzyme changes its shape to accommodate the substrate. Right? So an enzyme's function depends on its structure. If the shape of the active site is changed, the enzyme no longer functions, right? And so that is why you want to make sure that your enzymes are optimally working, right? They're working as best as they possibly can. And so enzymes function best in a smaller range of conditions, right? So it makes sense. Our different animals and plants have evolved in different environments. So what works best in the cold for one plant or one animal may not work best in like a hot environment like the desert, right? So enzymes function best in a small range of conditions. Changes in temperature. So this is what I was talking about. These are the conditions, either temperature or pH. It can break down the hydrogen bonds that change the enzyme shape. And so that's what happens when you cut yourself and you get some lemon juice or orange juice or any kind of citrus juice in your cut. That's an acid, right? And so when acid gets in there, it's going to actually change the pH of your proteins that are inside your skin, and it can mess those things up, those the hydrogen bonds. And that's kind of what an acid is. It just releases a lot of hydrogen bonds or a lot of hydrogen ions, which is hydrogen with a plus positive charge. But anyways, it when you cut yourself, you release acid into the proteins in your skin, and then the acid from that lemon juice messes with the hydrogen bonds in the proteins, and it hurts, right? It's kind of a painful experience. And so the same thing with temperature. If you have an egg and you boil that egg will no longer be able to carry out the functions of something living, right? So a chick will never hatch from an egg that has been boiled because you have denatured or you changed the protein. And let me actually put that word here because that is a very important word to know. When you use acid to change the shape of a protein or you use heat, you denature the protein. Right. So this word right here, I'm trying to kind of bring it up and yeah, there we go. So this word right here, to denature, kind of means to change the shape of an enzyme. You can either do that with temperature, the way you would in an egg, or you could do that with pH, the way you would with acid, right? And so I don't know if you all have ever heard of ceviche, but it's this dish where you kind of just soak raw fish in lime or lemon juice, and it kind of cooks it. It makes it firm and solid, almost like you cooked it with heat, but it's done with the acidity of lemon or lime juice. So it's very good, but it's very interesting how it denatures that fish protein when you add that lemon juice to it. All right. And like I said, y'all, some things work better in hot temperatures for animals or plants that are adapted to hot temperatures, and some things work better in cold temperatures, right? For plants and animals adapted to cold temperatures. But also think of pH, right? The things in your stomach have to be able to be very acidic. They should be able to withstand very acidic environments because there are a lot of acids in your stomach that break food down, right? So the enzymes that work there have to have a high tolerance for acid for acidic environments as opposed to enzymes in other parts of your body, right? So even within your body, you have enzymes that can take different pHs. Inhibitors also affect the way that enzymes work, right? And we'll see what that is right here. There's a couple examples of these inhibitors that we're going to talk about. And so when you're talking about a competitive inhibitor, they're just chemicals that resemble an enzyme's normal substrate, right? So they kind of look like what would normally go in here, but they compete, like it says right here, with it for the active site. So a lot of times when your body needs to produce something, once it's produced enough of those things, then a competitive inhibitor will come in here and basically stop more of these products from being created because you have enough. I don't remember if we've gone over homeostasis yet, but that's basically saying that your body has to have some balance, right? Your body can't get too acidic, or you're going to get sick. Your body cannot be too alkaline or basic, you're going to get sick. You get too hot and it's not going to go well. You get too cold and it's not going to go well, right? In order to have that to be living healthy, you got to have that homeostasis where your body has that balance, right? So if you create too much of something, this can competitive inhibitor comes here and it's like, all right, we have enough. I'm gonna just kind of hang out here, and once we run out, then I'll kind of pop off and we can continue whatever's was going on here before, right? But for now, it's just gonna take up this active site, so nothing else can come in here and be created. No more products, right? So again, the substrate is kind of blocked. So we have non-competitive inhibitors. And what these guys do right here is, you see right here, this is a non-competitive inhibitor. And so here you would have the active site normally open like this, right? So it's kind of got that opening right there in the active site. But with this non-competitive inhibitor is it comes and it attaches itself right here. And when it does this, it ends up affecting the active site. So now this active site is not open for business. It has been closed down so that nothing can come into that active site and no more products can be created. Right. So inhibitors that do not enter the active site, they bind onto another part. So like back here, this part of the enzyme, and it causes the enzyme to change its shape, right? Which in turn alters the active site. So again, nothing can come in here. And that happens again when there's already enough of these products kind of floating around in your body and there doesn't need to be any more created. So then this non-competitive inhibitor kind of shuts down the factory for a little bit, right? It's like we created enough product, we're overflowing at this point. So let's shut it down until we need some more, right? And then it leaves and the active site opens back up. Yeah, substrate has nowhere to go because the factory's closed for business, right? And so effects on temperature and so again, this is just saying right here that enzymes work really well. So this plant or animal, this organism, right, this living thing, is its enzymes are working really well around 40 degrees Celsius, right? That's why it's at the highest point, because this is basically saying this is where they're working at their best, right? If they're over here at like 0 degrees, it's too cold for these enzymes and they are not doing as well as they could be. Once it gets too hot, like a lot of times when you get sick, this is kind of what happens is some of your enzymes start getting denatured or changing their shape because of how hot your body gets, right? So like once you get to a certain temperature, especially if you get really overheated, your enzymes do not work very well, and so production goes down, right? So this is what it's saying is that temperature has an effect on enzymes, and it's not all the same for one living thing, right? Some living things do better even within a person, right? Some enzymes work better in acidic environments and some and not such acidic environments. So here you're seeing the temperature affects that, right? So around right here, what is that, like about 35 degrees Celsius is where they're working at their best, and 10 not very well, and 50 not very well, right? Because the rate of reaction, how fast it's going, is very low right here and very low right here, but it's real high up here when it's at about 35 degrees, right? 50 not very good, 10 degrees not very good. Same thing with pH, y'all. When something is over here in this area, it is said to be um, acidic. And when it's over here, it's said to be alkaline or basic, right? But right here in the middle, this is neutral. So it seems like right here where it's kind of neutral, this is where the enzymes are working the best, right? So probably not in your stomach, because those enzymes would probably be working best in these more acidic environments. Because again, you have acids in your stomach, they break down the food, right? So this is probably happening in a place where you don't need a very acidic environment, right? You need more of a neutral environment.